We hope that you'll enjoy this seminar. And today we have Mark Fleming speaking to us. I want to take 1 minute to share my screen to remind you about the Centers of Excellence in Hematology. This is the homepage for it. And what I really want to draw your attention to is a call for new funding, which you can find right here, hopefully, which has new funding opportunities, Type A awards, which are ongoing awards, where you can get credits to use cores at any of the core facilities around the country. And the information about that is right here. And then a second type of funding award, which is really focused on innovative pilot research in non-malignant hematology. And we're trying to focus on individuals who might be new to the field, who have a new project, and to potentially have trainees and fellows who would apply for these kinds of funding opportunities. And the details are here on the website that you might go and look at. You apply for it right here. This is a list of previous individuals who've received funding. And I highly encourage you to go look there to see if there's an opportunity that might be applicable for you, especially those of you who are here who are trainees. And now I want to introduce Dr. Fleming. And the first thing I want to tell you is I think I got introduced to Mark, golly, I don't even know how many years ago, a long time ago, but I'm going to tell one embarrassing story for myself. I had to give a talk at a BioIron meeting for a postdoc who couldn't attend. And I got her slides and got up to present on mitoferrins. And it was a 15-minute talk, rushed through the talk, and then had a good laugh that Mark was able to point out that we had the red pulp and white pulp on the spleen mislabeled, which was an awesome and embarrassing piece of information. But one of those pieces that it's like when students complain about how you teach, you never forget. Anyway.
Only the pathologist.
I know, but wow. After looking at that, I just thought, okay, that's terrible. Anyway, Dr. Fleming got his undergraduate degree at university. I'm going to ask you, if you're not muted right now, please.
Hey guys, you're mute.
Let's see, there we go. And please remember to mute yourself. Dr. Fleming got his undergraduate degree in molecular biology from Princeton University and his doctorate in organic chemistry at the University of Oxford in England. He then went on to Harvard Med School, where he got his MD, and he did his residency in anatomic pathology and a fellowship in hematopathology at Brigham and Women's. He also did a postdoctoral fellowship in the Division of Hematology Oncology at the Boston Children's. And he was named a Pew Fellow in Biomedical Research. And he is now the chief resident, what do you call it? Chief
of anatomic pathology.
Chief of anatomic pathology. That's a busy role. And as we talked before we started this, it's a busy role, especially in this time of COVID. And we really appreciate you being willing to talk in this virtual seminar series. I'm going to ask people to, for sure, mute themselves. I will monitor questions in the chat and invite you to ask your question. There's a natural break in the middle if you want to hold questions, but if it seems like it's a burning question, then we'll bring it up to be asked at that time. And with that, Mark, we'll let you attempt to share again.
Okay,
make sure we're up and running.
And hold on, let me share my screen and get going. All right, and we should get going right now. Does it look good?
Yep, it looks great.
Okay, so thank you for having me, Diane, and Iron and Heme community. I'd originally entitled my talk to focus sort of more broadly on sideroblastic anemias, but I thought I'd do a little experimentation today. This is a largely new talk. I'm going to focus on the heme-related sideroblastic anemias and some work we've been doing both in humans and in mice on the congenital sideroblastic anemias due to heme biosynthesis defects. So So the sideroblastic anemias are, broadly speaking, a group of anemias that all share a common morphological finding, and that is that they have erythroid precursors with abnormal iron accumulation within the mitochondria. And these appear to ring around the nucleus of the cell, giving rise to the name ring sideroblasts. Like almost anything, almost all diseases, there are acquired forms and congenital forms. Acquired forms of sideroblastic anemia are the most common. They're typically characterized by large red blood cells, or they're macrocytic. Most commonly, these are clonal disorders of hematopoiesis termed myelodysplastic syndromes, but there can also toxins, drugs, and metabolic causes can also instigate an acquired sideroblastic anemia. They have a very diverse pathogenesis that I'm not going to go into today. And notably, in 2011, it was found that about 80% or more of cases of the myelodysplastic form of sideroblastic anemia, refractory anemia with ring sideroblasts, about 80% or more of these cases are due to somatic mutations in a spliceosome factor, SF3B1, which largely to this date is not actually implicated in any way, shape, or form in the pathogenesis of the congenital sideroblastic anemias, which are the focus of the talk today. The congenital sideroblastic anemias are inherited mitochondrial disorders. They don't always present in infancy, as many congenital disorders do. Sometimes they present in later adulthood, as you'll see later today. These are relatively rare diseases. There are fewer than 1,000 cases reported. And they can be characterized by red cells that are small, microcytic, normal in size, normocytic, or macrocytic. They can be syndromic, and that is that they can also affect other organ systems, most commonly the nervous system, brain, muscles, but also the liver. And there can also be a variety of dysmorphologies, immunodeficiency, and a wide other number of other syndromic features associated with some of them. It's now clear, now that we know about the genetics of many of these disorders, that they're really due to a restricted subset of pathogenesis. And that involves diseases that are involved in heme biosynthesis, iron sulfur cluster biogenesis, and if you want to think of them as the same thing, as mitochondrial translation and oxidative phosphorylation or respiration. So if you want to look at the mitochondrion from a sideroblastic anemia biologist perspective, we have these four main pathways that I alluded to here. First, there are the heme-related sideroblastic anemias, X-linked sideroblastic anemia, which is the most common form of sideroblastic anemia, and SLC25A38 associated anemia. And these are going to be the focus of the talk today. Just to go through the other pathways, there's iron sulfur cluster biogenesis sideroblastic anemias that are mostly involved in the transfer of the nascent iron sulfur cluster, a complex involving HSCB and HSPA9 that transfers nascent iron sulfur clusters to enzymes within and outside of the mitochondria, as well as defects in oxidative phosphorylation and in mitochondrial translation. And since the mitochondrial genome encodes both tRNAs that are required for translation and several of the structural genes for oxidative phosphorylation in the mitochondria, really these are kissing cousins of one another. One disease, thiamine-responsive megaloblastic anemia, is likely due to multifactorial causes involving both tRNAs, the citric acid cycle, and iron sulfur cluster biogenesis. So the substrate for our work is a registry or a consented collection of specimens from sideroblastic anemia and iron disorders patients. The original collection started in approximately 1992, but beginning in about 2008, we began collecting sideroblastic anemia patient samples in earnest. Just for reference, prior to us starting this registry, there were really only two forms of sideroblastic anemia that were defined genetically. One was due to mutations in ALAS2, which we'll go into in the last part of the talk. There are also mutations in mitochondrial DNA or deletions associated with Pearson syndrome, but otherwise it was sort of a genetic wasteland. Early on during our work, other groups identified mutations for mitochondrial myopathy with lactic acidosis and sideroblastic anemia. As I already alluded to, patients with MDS, refractory anemia with ring sideroblasts, could be excluded genetically in 2011. So as we began to collect these, we began to collect patients that by and large did not have mutations in any of the known genes, particularly not ALAS2, nor did they have mutations in mitochondrial DNA. To this date, we've now collected 251 probands from sideroblastic anemia families. Shown on the right is the breakdown of the fraction of these families. We can now explain nearly 3/4 of all these patients. Using both positional cloning and exome sequencing, we've published 7 novel genes, and in our exome escapades have led to a discovery of 13 other unpublished genes that I'm not going to talk about today, but are listed here in the pie diagram. The most common of these can only count for 2.4% of all of our patients, so about 5%, 5 patients in our group. So let's go back and focus on the 2 heme-related sideroblastic anemias. First is X-linked sideroblastic anemia, which is due to mutations in ALAS2, which is the rate-limiting and first committed step in heme biosynthesis, as well as in SLC25A38, which we now know to likely be a glycine importer necessary to import glycine, which is one of the substrates for ALAS2. So the story of SLC25A38 sideroblastic anemia was one that began early on in our collection, which I inherited part of the collection from Sylvia Bottomley. And in that group, she had a number of patients that were very similar to X-linked sideroblastic anemia. They had small, pale red blood cells. They also developed secondary iron overload. But unlike X-linked sideroblastic anemia patients, which are often responsive to large doses of vitamin B6, which is the cofactor for ALAS2, these patients were uniformly not responsive to pyridoxine therapy. Furthermore, these patients they tended to be present very early on in childhood, often at birth, sometimes in utero, often in infancy, and they became transfusion dependent nearly to the one. And unlike X-linked sideroblastic anemia, females were equally affected from males. Most notably, we had several autosomal recessive pedigrees that included both males and females in the family, and several of these families came from consanguineous marriages, suggesting once again that it was a rare recessive disorder. About the time that we were getting set to actually to try to clone this gene, I was contacted by Mark Samuels of the Atlantic Genome Canada, Anemia that were not thought to be related to one another directly, or at least through a direct lineage, but all came from a region in northern New Brunswick in the Baie de Chaleur region, which is one of the ancestral Acadian founder populations. French Canada, as you may know, was settled by perhaps 100 or so families. And so many of these families are ancestrally related to one another. So Mark and colleagues took the idea that this could actually be that all of these families that he had were actually related to one common ancestor. And using what was then current single nucleotide polymorphism homozygosity mapping, they were able to map this disease gene to about a 3 megabase interval containing 32 genes, which in the recently completed draft of the human genome, there is one really obvious candidate gene within the region. And that gene was SLC25A38. SLC25A38 is a mitochondrial carrier family member. These are proteins that usually function as inner mitochondrial membrane importers or exchangers. This particular one was expressed nearly exclusively in erythroblasts, particularly in late erythroblasts when heme biosynthesis was maximal. And of these SLC25 members, there are different members that transport keto acids, adenine dinucleotides, and amino acids. SLC25A38 was a member of the amino acid carrier family. And when Paul Schmidt in my lab knocked out the yeast equivalent of this, he was able to show that in fact the yeast deficient in SLC25 or A38 ortholog had an oxidative phosphorylation defect that was due to heme deficiency. When our collaborator Mark Kellogg measured ALA, aminolevulinic acid, the heme precursor in these cells, aminolevulinic acid was deficient. And then when you supplemented these yeasts with either ALA or glycine, one was able to correct the defect, suggesting that SLC25A38 was likely the glycine importer required for high-level heme biosynthesis within erythroid cells. Subsequently, another group has gone on to show in yeast proteoliposomes that SLC25A38 does indeed mediate high-affinity glycine uptake into mitochondria. So over the course of years, there have been 92 families with SLC25A38 mutations reported in the literature. And within our registry, we've collected 36 families, and they include 49 patients. In a recent paper published in Human Mutation, we were able to elaborate very definitively on the clinical features of disorder. Almost all patients present within the first week of life. Some are transfusion dependent in utero, and it's extraordinary for a patient to present after the age of 4. These patients have very low hemoglobins, a normal hemoglobin being in the 12 to 15 range. So they have a hemoglobin that's less than 1/2 to 1/3 of normal. They have very tiny red cells, and both of these are typically more severe than what one sees in X-linked sideroblastic anemia. Nearly every patient is transfusion dependent. Some have gone on to be transplanted and fully correcting the disease. But many of these patients actually have diverse syndromic features. And it's not because we believe that the SLC25A38 anemia is itself syndromic, but it's more the fact that, like many rare recessive diseases, these disorders are most common in consanguineous marriages. And consequently, it's more than likely that this very diverse other syndromic features are probably due to other recessives segregating in these families. And the entire population of patients, there are now 47 described mutations at 40 codons. And these are distributed more or less equally between missense, frameshift, and stop-gained mutations, if one includes, and with splicing mutations and stop-loss mutations being much more infrequent. Very interestingly, almost all of the missense mutations occur within a transmembrane domain. And interestingly, transmembrane domains are not particularly well conserved compared to other portions of the protein. So it appears that these missense mutations nearly exclusively occur in transmembrane domains and likely disrupt transport function and/or the stability of the protein. Furthermore, it's interesting that the mutations that occur are not necessarily at conserved amino acids, rather that they tend to recur at CpG dinucleotides, which due to methylation of this methyl C, which can be deaminated to form uracil and then is corrected to thymidine, result in transition mutations in the DNA. So as I show here, here's the distribution of mutations between frameshift, missense, splicing, and stop mutations. As I alluded to, a large fraction of the patients are homozygous. Many of these patients, while they're not known to be consanguineous, like the Bay-Chaleur patients, they come from isolated populations that are presumed to be genetically restricted. And very interestingly, more than half of the patients are likely nulls or near nulls, suggesting that there are other ways to get glycine into the mitochondria for heme biosynthesis. So we went on to model SLC25A38 in the mouse. The SLC25A38 null animal, unlike the SLC25A38 null human, is actually lethal. And if one looks at the embryonic erythroblasts that actually circulate in the peripheral blood of these animals, one can appreciate that they have massive inclusion of iron within mitochondria of these cells, of even the embryonic erythroblasts. In order to get around, circumvent this lethality, we actually crossed it to another animal, the VAV-Cre. VAV-Cre is expressed in all hematopoietic lineages, but it tends to turn on a bit later than other Cres. For example, EPO-Cre turns on a bit earlier. So consequently, we actually observe live-born animals in the VAV-Cre animals. Here on these iron stains, you can appreciate that the red blood cells are pale and they're smaller than their counterparts. So this is the hypochromic microcytic anemia. And under very high power, you can appreciate on an iron stain that there are lots of iron positive granules that are electron densities on transmission electron microscopy corresponding to iron deposition in mitochondria. But unlike humans, mice don't seem to develop ring sideroblasts, perhaps because of much of the heme that's made in a mouse erythroid cell is synthesized after it is enucleated. So we do not see ring sideroblasts in the bone marrow, but we do see abundant what we call siderocytes in the peripheral blood. We look at the quantitation of the anemia. These animals have a hemoglobin of about 10, so they're not as severely anemic as the human patients. They have very tiny red cells with a very wide red cell distribution width. And unlike humans, and typical of many mouse anemias, they actually have a high reticulocyte count. Humans typically have a low reticulocyte count or hypoproductive anemias. So we wanted to understand, using this animal, could we treat the anemia by providing excess substrate? So first of all, we tried glycine, but we also tried serine. And to go back to a bad dream, for some of you may recall, that glycine and serine can actually be readily interconverted from one to the other within the cell. First, serine hydroxymethyltransferase pathway can convert serine to glycine in the cytosol. And similarly, the glycine cleavage pathway can actually generate going in reverse, can actually generate glycine from serine within the mitochondria through a mitochondrial ortholog of SHMT1 called SHMT2. And that glycine can then go on to become the substrate for ALAS2 and ultimately heme biosynthesis. So Anup Sendamarai in my laboratory fed SLC25A38 VAV positive animals, and we only treated the mutants alone in this with either a basal synthetic diet that contained 1% glycine, which is typical, and 0.35% serine, which is typical of most sort of natural mouse diets. A diet that was high in glycine, or about 10% glycine, and this is actually the amount that's actually tolerated by humans. Glycine is actually a relatively sweet substance, and we can eat diets up to 10% with no adverse effect. And we also did the reverse diet, which was 10% serine. We fed the mice for 16 weeks, and every 4 weeks did complete blood counts. And although the headings are rather small here, what we were able to show is that actually glycine did nothing for the hemoglobin. In fact, the hemoglobins of the animals fed the glycine diet were actually worse than the animals fed the serine diet or the basal diet. Very interestingly, however, the mean cell volume of the cells that were produced by the animals with the glycine were actually normal in size. And furthermore, although I'm not showing them here, the animals fed glycine, all of those siderocytes that were in the peripheral blood were in the peripheral blood disappeared. So it appeared that what happens with glycine feeding is that you actually correct the red cell size and hemoglobinization problem as well as the siderocytes but that there's perhaps a toxic intermediate that develops that suppresses erythropoiesis overall. And consequently, the number of red blood cells produced by the animals, shown here in the reticulocytes, is unable to compensate for the degree of the anemia. So the net is actually a more anemic animal that actually has normal red cells. So at this stage in the project, we really want to try to understand, where does the glycine from heme synthesis come from in the absence of SLC2538? Is amino acid supplementation a potential treatment for this disease? And is there some inhibitor or a byproduct that develops by feeding excess glycine that actually suppresses it? Or does it require other cofactors such as folate, which is involved in the glycine shunt and is used by serine hydroxymethyltransferase? So at this point, this is still a work in progress, and we're looking forward to doing some protein biochemistry as well as metabolomics using mouse models as well as cell line models in order to look at this further.
Mark?
Yes.
I just have a question. When you do the glycine feeding or the serine feeding, do you note any other amino acid shifts, like the necessity of getting glycine into the mitochondria makes it so you skew your amino acid homeostasis for other amino acids?
No, so when we looked at the global plasma amino acids, we did not look at the red cell amino acids. But if you look at the global plasma amino acids, there's no difference other than some slight differences, other than the fact that the amount of glycine in the plasma is 8 to 10-fold that in a normal in the basal diet. So we haven't really exhaustively gone to see if, for example, pushing glycine into some other toxic metabolite pathway that accumulates, or whether we precipitate some other amino acid deficiency. These diets are in fact nitrogen balanced for one another, which is one concern that we had. But your question is in fact a good one.
Thanks. Any other questions? Not right now in the chat.
All right, great. So let me move on to X-linked sideroblastic anemia. X-linked sideroblastic anemia is the oldest of the sideroblastic anemias. It was originally described by a man named Thomas Cooley. And some of you may know this name because Cooley's anemia is beta thalassemia. So this is Cooley's other anemia that he originally described in 1945. It was recognized by Rundles and Falls the year after that this was a hereditary disorder. And then somewhat later that this anemia was uniquely responsive to high doses of vitamin B6. So it became known as pyridoxine responsive anemia. The term sideroblastic anemia wasn't coined until 1965 at a consensus conference to try to make heads or tails of these disorders. So X-linked sideroblastic anemia is the most common form of sideroblastic anemia. In our cohort, it's about 30% of patients, but it probably represents 75% of all the sideroblastic patients described in the literature. It presents in males, typically from age 1 to often greater than 60 years of age. I've diagnosed 70 to 80-year-old men with X-linked sideroblastic anemia with small red cells and iron overload as the only features. They don't actually have anemia. Classically, these males typically present at age 9, 10 to 15, often in peripubertal years when there's some hematopoietic stress. It's a microcytic hyperchromic anemia with small, pale red cells of varying size and shape. About 1/3 of patients, if you give them up to 200 milligrams of pyridoxine, which is about 100 times the recommended daily requirement of pyridoxine, you can raise their hemoglobin into the normal range. About 1/3 of patients get a partial response, and 1/3 of patients have no response at all to pyridoxine. And these are the pyridoxine non-responsive patients. Not uniformly, but very commonly, these patients would present with secondary iron overload, even in the absence of transfusion. This is one of the classic iron-loading anemias, and as you might expect, both ERFE is induced and hepcidin is suppressed, though I'm not going to show you that data today. As I alluded to before, these are mutations in ALAS2. And by and large, these are missense mutations, unlike SLC25A38. These are unstable enzymes that are many of which are stabilized by vitamin B6, and they're often due to substrate binding defects, dimer interface problems, or folding problems. In this disorder, about 5% of patients have a mutation in an exon 1, GATA1 transcription factor binding site in an enhancer element. There are rare promoter mutations. There are very rare splicing mutations. We just described the first splicing mutation in a male patient with a synonymous coding variant that actually resulted in a splicing defect. And nonsense mutations are seen in women only because ALAS2 is an essential gene. So amongst our collection of sideroblastic anemia patients, we have about 75 X-linked sideroblastic anemia patients. There are 230 some odd males described in the literature, as well as those that we have collected. If one looks at the distribution of mutations, and this is, so the mitochondrial targeting signal is encoded in these exons here that are not shown. Exon 5 here contains arginine 170. Exon 10 contains, exons 10 contain arginine 411, and arginine 452 is also contained here. And together, these three mutations constitute 40% of all the described probands in the literature and in our data set. Interestingly, these are not the most conserved amino acids in the protein. Rather, they are arginines that contain CpG dinucleotides, which, as I inferred in SLC25A38 disease, these are prone to mutation. Each of these mutations is somewhat different in its response to vitamin B6. Mutations at R542 are classically very B6 responsive. These patients can go from hemoglobins of 6 to hemoglobins of 12 to 15 on supplementation. Mutations at arginine 411 are variably responsive or non-responsive depending upon the particular mutation. And mutations at arginine 170 tend not to be vitamin B6 responsive mutations. We've been increasingly interested in X-linked sideroblastic anemia in females. So although this is an X-linked disease, there are a remarkably large number of women that have been described with the disease. There are about 40 families that are reported, and we in our collection have another 20 individuals from 18 families. The average age that women present with X-linked sideroblastic anemia is about 40, versus in our cohort, it's about 20 years of age. And unlike men, females with X-linked sideroblastic anemia tend to have macrocytic anemia. They tend to have very large red blood cells, often in the 100 femtoliter or more range. And unlike men, they're typically not pyridoxine responsive. In fact, the only females that are pyridoxine responsive are the rare females that have microcytic anemia. Because they present with macrocytic anemia at an older age, they are often mistaken for patients with myelodysplastic syndrome. And myelodysplastic syndrome, but in the age of mutation analysis for SF3B1 as other somatic mutations, it's becoming increasingly apparent that this misdiagnosis is very common. And this is actually a germane thing because an MDS diagnosis carries a potential of leukemic evolution, whereas X-linked sideroblastic anemia is not associated with an increased risk of leukemia. So shown on the right is the sort of the breakdown of the cases. We've seen about 2.5 times as many males with ALAS2 as females, and about 30% of our probands. Again, probably biased to the fact that if a male had X-linked sideroblastic anemia, they wouldn't get referred to us, whereas a female might, because people don't think of the diagnosis. The mutation spectrum that one sees in X-linked sideroblastic anemia in females is somewhat distinctive. So one still sees the mutations at arginines 170 and 411, but essentially absent are mutations at arginine 452, which are the pyridoxine responsive ones. We also see a relatively common mutation at arginine 204 that creates a stop mutation, and another mutation at arginine 227. Once again, these are all CpG dinucleotides that result in these mutations. And together, these 4 mutations, these 4 different amino acids constitute 40% of probands. Interesting, many of the mutations other than those at R170 and R411 are mutually exclusive from mutations that occur in males. And more often, these mutations are conserved amino acids. And as I alluded to, they're typically not vitamin B6 responsive. So here's the hematology of the patients that we have. So X-linked sideroblastic anemia females are comparably anemic to the males. If you look at carriers of X-linked sideroblastic anemia that are not clinically affected, and we define clinically affected as having a hemoglobin of 11.5 or less, they have essentially normal hemoglobins compared to their wild type female relatives. As I alluded to, the mean cell volume in females with X-linked sideroblastic anemia is typically in the mid-90s or thereabouts with a very wide range, whereas in males, it's microcytic, often profoundly so. And if one looks at the RDWs, the red cell distribution widths are typically very high in males, less so in females, but the carrier females, the RDWs, are moderately increased, which leads to the observation that many times if you look for the red cell distribution in the mother of an affected male, that you have a clue to the diagnosis of X-linked sideroblastic anemia, because this dimorphic pattern of red cells is typical both in the female carriers and in the affected males. Interestingly, however, if you break out the affected females into groups of females that have mutations that have also been seen in male that are affected males versus females that only have mutations that are described in females, there's a difference in the phenotypes. Very strikingly, the females that have mutations that are only observed in females have more severe anemia, and their mean cell volumes are often much more at the extreme, typically much higher than the ones that have mutations that are expressed in male. So what is the cause of this? So why is it that we have a subset of carriers that are affected, a subset of carriers that are unaffected, and that there seems to be this difference in the clinical expressivity depending upon what type of mutation the patient has, that is, whether it's been seen in a male before or not. So we need to go back to the biology of X chromosomes. So as we know, in humans, the X chromosome in females undergoes random inactivation. And if you look at the X inactivation ratio using a polymorphism in the human androgen receptor of a group of infants, it's a normal distribution separated over at 50%. And if you look at 2 standard deviations, or about plus or minus 30% from 50%, so between 20 and 80. There are very few outliers in the infant population. But as it turns out, for a number of reasons, and it's not due to the acquisition of mutations in clonal hematopoiesis genes that are associated with myelodysplastic syndrome, women, as they age, can skew their X chromosomes in one direction or another. Now, this is a very subtle difference between clonal, because clonal, we sort of ascribe to a mutagenic event that gets propagated through all of the cells from that daughter. Whereas X chromosome inactivation is an epigenetic phenomenon that they can all have a similar epigenetic pattern, but they're not necessarily clonally related to one another. If one, however, takes a large population of unaffected females from our database of a wide variety of ages, you can appreciate that the distribution is still normal, but that it's flattened. And now that there are females on either side that have skewed as they've aged. So this likely accounts for why we see X-linked sideroblastic anemia presenting in older females. And it's also the fact if you think about it, the skewing probably occurs at the stem cell level and not at the erythroblast level. Because if it occurred at the erythroblast level, there might be selection against that. If it occurred at the erythroblast level, there might be selection against it. So there's probably a very early event that leads to this skewing. So what does the X chromosome inactivation ratio look like in females with X-linked sideroblastic anemia? As you'd expect, most of the patients are outliers. The gray box shows the two standard deviations away from the mean. But it's interesting. There are females who are skewed toward the mutant chromosome who are not clinically affected. And as it turns out, and I'm not going to show you this breakdown, these females more often have a mutation that's commonly seen in males. And so the implication is it's likely that the mutations that result in this disease are more often more severe. Interestingly, there are, however, some females who appear to be not skewed, at least not skewed, and maybe even skewed away from the affected X chromosome. So this could be either a crossover event between the androgen receptor and ALAS2, which are actually closely linked to each other. Or it could be that there's somehow some other epigenetic phenomenon that's going on that we're not aware of, that leads to allele-specific expression by another region. Now, the X chromosome inactivation ratio is done on peripheral blood leukocytes. So the X chromosome inactivation is measuring what is the state of expression of the white cells. In order to measure the relative expression of the ALAS2 allele, one has to do RNA sequencing on peripheral blood of the ALAS2 mutation itself. And here we have many fewer individuals, but I think you can get the picture that this 50% right there would be half and half normal and half mutant. So in the affected females, consistent with the skewing of the white cells, they're pushing out more of the mutant of the mutant red cells, but they're still actually a minor fraction of all of the red cells throughout in the peripheral blood. And those females that are unaffected essentially have suppressed all of the or many of the red of the expression of the production of red cells with the mutant X. So here, what we're looking at is actually two different populations. We're looking at most of the red cells, or most of the white cells in an affected female with X-linked sideroblastic anemia derived from the mutant X chromosome, whereas most of the red blood cells derive from the wild type X chromosome. And therein lies why females with X-linked sideroblastic anemia have large red cells. So it's likely that the females have normal red cells that have a normal MCV, and that some of them have stress hematopoiesis, which tends to give you a higher MCV. And it's not due to reticulocytosis. So this explains why there's this paradoxical change in the MCV between a male with X-linked sideroblastic anemia and a female with X-linked sideroblastic anemia.
Mark, we have a question in the chat from Joe Percal. Joe, do you want to unmute yourself and ask?
Oh my God, this is going to be a bad one. Tough one. Okay, Joe.
I'm totally innocent. Can you comment on the molecular basis or cellular basis of dimorphic morphology in sideroblastic anemias?
Yeah, so it's interesting. It's always said that it's the females that have dimorphism, but the males have dimorphism.
Of course, yeah, I've seen.
Okay, so what I think is actually happening is I think that it's kind of a stochastic event. That what happens is that you're getting iron accumulating in mitochondria, even in the males. And so in the females, you can explain dimorphism from the X inactivation. In males, the dimorphism, I think, results from the fact that you get some erythroid progenitors whose mitochondria poop out really early, and they don't make much hemoglobin. And there are other red cells that kind of get through to later cells, and they produce more normal red cells. And in fact, when you look in the peripheral blood of men and you stain them for iron, the siderocytes are always the microcytic cells, and the normal cells are the macrocytic cells. So I think it's just a relative effect of when that red cell decided to enucleate and how much hemoglobin it had actually made based upon how much its mitochondria were affected. As I'll show you, we see that in the mice too. So it's probably I can't think of any other hand-waving explanation. Can you?
No, I think certainly it makes sense, but as you said, it's virtually universal, right?
Right.
In both men, right? And if I may make another comment on X chromosome inactivation, when you use the androgen methylation, you need to have a DNA, right? But there are actually much more biologically more sound assay based on transcription polymorphism, exonic polymorphism of X chromosome, and you can do it in reticulocytes, right? Because you have an RNA. And so you could probably focus much better on the selection of the X chromosome marker that way.
Yeah, we definitely could. But in this case, since we have an X chromosome marker, right, with the mutation, that's what we chose to follow.
Sure.
In the females. Okay, good questions. So what are the challenges of studying X-linked sideroblastic anemia? First of all, it's modeling terminal erythropoiesis in vitro. Ring sideroblasts are the most abundant in the latest erythroblasts in the heme-associated sideroblastic anemias. But primary erythroblasts don't often mature all the way out in culture. There are better cell culture systems. And furthermore, until recently, even primary erythroblasts couldn't be developed into ring sideroblasts in culture. There are now methods actually that you can use iPSCs or HuDep2s that will develop ring sideroblasts in culture, but they involve sort of contrived conditions and lots of extra iron. Also in the field, the ALAS2 knockout's been attempted, but it's lethal as one would expect because it's an essential gene. Furthermore, the mouse GATA2 binding site deletion in ALAS2 animals are lethal. And as I alluded to before, mice don't develop ring sideroblasts in any sideroblastic anemia model, except for one sideroblastic anemia model where a human ALAS2 was expressed at low levels in the mouse knockout. There are, however, abundant siderocytes in the peripheral blood, so we still think it's a good model. So Sarah Ducamp, who is a postdoc in my laboratory, who's now out on the job market and presenting a poster at ASH in 2021, decided that she wanted to model X-linked sideroblastic anemia in the mouse. And she made those 3 common mutations present in men, R170H, R11H, and R452H, which, as you recall, were a variety of severities and a variety of different responsiveness to pyridoxine. I'm going to focus on the R411H animal today because it's the most severe, but Sarah's poster will describe many other phenotypes in these animals at ASH. So here in blue are wild type mice fed a normal chow diet, which contains about 16 part per million pyridoxine, which is about 2 times the RDA of pyridoxine for a mouse. The normal animals, here's a normal hemoglobin. Somewhat surprisingly, the R170H mice have only a mild anemia. The R452 mice have very little anemia at all, as would expected. But the R411H mice have a very low hemoglobin, a very low MCV, and a very wide RDW compared to the other mutations. When we look in the peripheral blood, we see the characteristic siderocytes. Here you can see that the siderocytes are, Joe, to your question, the siderocytes are in the most hypochromic cells. So this is sort of the dimorphism, if you will, here, revealed on the iron stain. And by transmission electron microscopy, you can appreciate that the mitochondria are more electron dense, typical of iron deposition within the mitochondria of the reticulocytes shown here. If you look at the bone marrow, or if you look at splenic erythroblasts, there's a relative defect in maturation from the early erythroblast to the late erythroblast, and then ultimately to the mature red cell here in the mutants compared to the wild type animals, even on the baseline diet. But what's really remarkable is when you make these animals vitamin B6 deficient. So in order to do this, what Sarah did was she allowed animals to be born. At age 3 weeks, the animals were placed on a defined vitamin B6 diet. And then every 2 weeks thereafter, for the next 8 weeks, the animals, she obtained a CBC. And so we're going to show you the time course of that phenotype over time. So here's the R411H animal. You notice that a wild type animal, even on no added pyridoxine, so no pyridoxine in this synthetic diet, is relatively resistant to the development of anemia, though it does have siderocytes in the peripheral blood here. Whereas the R411H animals develop anemia very precipitously, and by 8 weeks on diet have a hemoglobin of about 2 and need to be humanely euthanized because of their profound anemia. At that time, they have massive splenomegaly. Their spleens are about, this is a log scale, they're about 80 times as large as a normal animal, and they're jam-packed with proerythroblasts, which, as you might expect, there's even a more profound defect in maturation here than when they're fed the normal chow diet. Now, if you look at the increasing the pyridoxine levels from 0 to physiological to pharmacological, one can see that there's actually a correction of the erythropoietic defect going from when one gets to the highest level of supplementation. And here you actually see a very slight increase in the hemoglobin in these animals at the B6 dose of 300 part per million. So this is still an unfinished story that Sarah will move on to finish herself, but we're interested in other questions that we can ask with this. So what's the role of post-transcriptional regulation of ALAS2 by IRPs? So for example, a number of other sideroblastic anemias, the dysregulation of ALAS2 is thought to play a role in that pathogenesis. Is it possible that we could somehow modulate ALAS2 expression in these ALAS2 mutations by targeting the IRP to knock down the iron-dependent regulation and free the ALAS2 from IRE-dependent suppression. Are there other potential treatments for X-linked sideroblastic anemia? Iron deprivation, either through hepcidin mimetics, iron-deficient diets, or TMPRSS6 siRNAs has shown some efficacy in other mouse models of microcytic anemias. Is it by limiting iron? Would we limit the development of ring sideroblasts and promote the maturation of red cells, ameliorating the anemia? Where's the response of erythropoietic stimulating agents, both the conventional EPO, prolyl hydroxylase inhibitors, or, for example, the activin trap ligand, activin traps that have been shown to work in both beta thalassemia, intermedia, beta thalassemia major, and in refractory anemia with ring sideroblasts. And we also hope, using a variety of biochemical and metabolomic methods, that we can get to what is the elusive mechanism of ring sideroblast development, and why should iron accumulation in the mitochondria be dysregulated by heme biosynthesis as well as other defects. So with that, oh goodness grief, oh, here we go. I'm gonna say I had the wrong slide. This is still the wrong slide. So the people that contributed to the sideroblastic anemia projects in my lab, I mentioned Sarah, who did most of the work with the X-linked sideroblastic anemia animals. Anup Sendamarai did the work. This is still my wrong acknowledgement slide. Anup did most of the work with the ALAS2 X inactivation and the SLC25A38 animals. They were ably helped by Dean Campagna and Paul Schmidt in my lab. Matt Heaney is my co-conspirator on the clinical end at Boston Children's. Sylvia Bottomley is the doyen of sideroblastic anemias, and she introduced me to this field. I, of course, have to thank the sideroblastic anemia patients and families. We received funding from the Manton Center at Boston Children's, the NIDDK, and ASH. And I also want to thank my clinical collaborators, including too many astute clinicians that have actually recognized these patients and sent them on to me this year. And also the Comprehensive Center for Excellence in Hematology at Boston Children's, Yuko Fujiwara in particular, who helped us design and execute the ALAS2 mutant mice. So with that, I'm happy to take any other questions.
Great. Thank you, Mark. What a great talk. Really, the female sideroblastic anemia and the X chromosome piece is really cool. Really enjoyed that. Does anybody have questions they want to ask? Feel free to unmute yourself and ask them.
Great talk, Mark.
Thanks, Joe. And you can ask me any more hard questions because they're hard. I really would like to understand what the basis of skewed X chromosome activation was. It's interesting that because there's some suggestion that there's actually familial X chromosome inactivation. I actually don't believe that's true. It's actually so common, and X-linked sideroblastic anemia is so common that I think it could happen by chance in families. But exist is actually very closely linked to ALAS2. And so we've looked for exist polymorphisms, but it turns out that exist itself is incredibly polymorphic, and it's not well covered on exomes. So you have to do it on genomes. So that would be the obvious candidate.
I'm still.
All right, Mark, so I will ask this.
Thanks, Iqbal, go.
So I really enjoyed it. It was a fantastic, almost felt like I was in a genetics course with Mark Fleming. Hey, Mark, so why is there a skewing going from an infant to an adult in terms of X inactivation?
So
that graph that you showed, yeah.
Yeah, so the mechanisms are not known, right? X inactivation is an epigenetic phenomenon, right? However, one hypothesis was that really that what the females are developing is clonal hematopoiesis of indeterminate potential driven by the same genes that are implicated in clonal hematopoiesis and ultimately in myelodysplastic syndromes in people. So you could think that you could acquire, for example, a somatic DNMT3A mutation in one cell that happened to have the X chromosome that was active was the mutant, and that cell propagated clonally through, right? In fact, when we've done exome sequencings on all of these females, 2 of them have clonal hematopoiesis. One of them is a control, and one of them is an unaffected female X-linked sideroblastic anemia character. So it's not clonal hematopoiesis. It probably truly is something of acquired X chromosome skewing in the stem cell population. And what and how that actually occurs is not entirely clear.
Wow. Okay.
Very interesting.
So how accurately looking at a white cell population reflects what's happening in the erythroid population when you're looking?
In the few people that have done it in bone marrow and separated out the erythroblasts are actually skewed themselves the other direction, probably because truly from selection from death, from cell death.
Right.
So then you extrapolate saying this is then inverse of what you would expect on the erythroid population versus what's happening on the myeloid.
So the myeloid population is not undergoing any selective pressure, whereas the erythroid population is undergoing selective pressure in the bone marrow, and that's occurring during maturation, right? So all the time that's in the bone marrow, and I bet it's even occurring after maturation because hypochromic microcytic red cells are abnormal red cells. They have shorter half-lives, and and who knows, some of these RNAs might be unstable, especially the null mutations. So those are going to get cleared faster. So it's probably, so I don't say that it's really, it could be it's the sum. What we're looking at the red cells is the sum total of all of those mechanisms added up. But
I think there is another theoretical possibility, and that is acquired uniparental disomy. And if it is so, there would be some cells in the female which would become homozygous, right? Similar to what happens with JAK2 mutation in polycythemia vera.
Yeah, but acquired uniparental disomy, we would actually see a single peak on the sequencing, and we see
Right.
2 peaks.
Right.
We see
But acquired uniparental disomy would happen only in some cells, in some stem cell, right? So there would be a population which would
but
have
it should be reflected in the peripheral. It should be affected in the peripheral blood cells too.
Well, yeah, that is correct, and you can do individual BFUEs and see any of them would become homozygous, right?
Right. I mean, yeah. Or
And it could be a relatively small population of cells, and you may miss it by the it should.
Well, if the red, I think you'd still see it in the white cells. It should be pretty obvious in the white cells in the peripheral blood.
Right.
I mean,
it should
you
be,
could have a small population, but that would be, you could see that by single cell sequencing, probably. Either single cell RNA-seq or single cell.
Or the BFU colonies, right? Colonies.
Yeah, but we never get marrows from these patients. They get one marrow.
But you can do it from peripheral blood, right? BFUs can be easily grown.
Okay, any other questions?
Very stimulating.
Yeah, a great
Thank
talk,
you.
Mark. Thank you so much, and thanks
All right.
everybody for attending. Want to remind you that our next seminar is not until January, and the speaker for that will be Adam Hughes, who's here at the University of Utah, whose title is Understanding How Cells Cope with Lysosome-Mediated Iron Deficiency. And hope to see you all there. That will be on January 11th. Thanks, and take care, everyone.
Bye.
Thanks again, Mark.